will start a new chapter about cells and cell physiology. We will talk about cells, cell structure, and methods of permeation through the membrane. All living organisms are made up of cells. This can be called the cell theory. Cells can be prokaryotic or eukaryotic. The prokaryotes are found in bacteria. They do not have a nucleus or cell organelles, and they don't divide through the process of mitosis. Eukaryotic cells have cell organelles and a nucleus and develop through the process of mitosis. These cells are found in plants, animals, and humans. Cell structure. It is composed of cytoplasm and plasma membrane. The cytoplasm of the cell is made up of a semi-liquid gel. The plasma membrane contains large amounts of cholesterol that stabilizes the lipids in the membrane, reducing its fluidity, and is made up of proteins and lipids. The globular proteins seem to float among the lipids, so the membrane has a fluid mosaic structure. The transmembrane proteins occupy the entire thickness of the membrane. They function as channels for the membrane transport of organic molecules. Glycoproteins are carbohydrate molecules associated with proteins located in the outer environment of the cell. Peripheral proteins are attached to the surface of the membrane. These are generally enzymes and have a role in remodeling the cell during cell divisions and contractions. Glycolipids and glycoproteins on the outside of the cell allow cells to recognize each other. They serve as receptors for signaling molecules, like hormones. Phospholipids are in a single double layer. Thus, we can say that the plasma membrane has a bilayered structure. They have a polarized end and a non-polarized end. The polarized end has phosphorus and is hydrophilic, meaning that it loves water. Therefore, it attracts it. The non-polarized end is made up of fatty acid chains and is hydrophobic, meaning it hates water, therefore it repels it. Thus we can say that the structure of the membrane is like a sandwich. This property of the phospholipids allows the plasma membrane to increase its surface area when Golgi vesicles fuse with it. Glycolipids are carbohydrate molecules attached to a lipid layer located on the outside of the cell. The plasma membrane is a semi-permeable membrane because oxygen, carbon dioxide, and water slip between phospholipid molecules. For other substances, there are other permeation methods, such as the passive transport, the active transport, and cytosis. Passive transport is done by diffusion and facilitated diffusion. Diffusion takes place from high concentration to a low concentration according to a concentration gradient. This does not require energy. For example, in the body, the diffusion takes place in the pulmonary alveoli, from which oxygen goes to the red blood cells in the veins. One type of diffusion is osmosis, which represents the diffusion of water molecules through a semi-permeable membrane from a region with a low concentration of a dissolved solution, which is also called a solute, into one with a high concentration. An example of osmosis is the reabsorption of water at the level of the renal tubules. You might ask yourself, why osmosis is carried out without energy despite the fact that it takes place in opposition to the concentration gradient? The answer is that in this situation I'm referring to the osmotic pressure which takes place from low to high. What you should always remember is that when the permeation through the membrane takes place from low to high concentration, it requires energy. Otherwise, it does not require energy. A solvate is a chemical substance dissolved in a liquid or a solvent. An example of a solvate is sodium chloride, and an example of a solvent is water. Now I will explain what happens to a cell when it is placed in a hypertonic, hypotonic, or isotonic solution. A hypertonic solution has a higher solute concentration than the inside of the cell. When the cell comes into contact with this solution, it shrinks or shrivels because water leaves the cell. When there is a lower concentration of a solute than in the cell leases and swells because water will enter the cell. In an isotonic solution, that is, in which the concentration of the solute is the same both outside the cell and inside the cell, no change occurs. Now let's continue with the membrane permeation methods. Facilitated diffusion takes place with the help of a carrier protein from a high concentration to a low concentration. Therefore, it does not require energy. The rate of diffusion depends on the number of carrier proteins. A great example is the diffusion of glucose in red blood cells. Active transport occurs when proteins transport chemical compounds across the membrane. It is carried out from a low concentration to a high concentration with the help of some transporter proteins. Therefore, it requires energy it procures from ATP. An example of active transport is the reabsorption of salts at the level of the renal tubules, or in the nerve cells when sodium goes outside the cell where there's already many sodium ions, and the rate of Now let's continue with the membrane permeation methods. Facilitated diffusion takes place with the help of a carrier protein from high concentration to a low concentration. Therefore, it does not require energy. The rate of diffusion depends on the number of carrier proteins. Great example is the diffusion of glucose in red blood cells. Active transport occurs when the protein transport chemical compounds across the membrane. It is carried out from a low concentration to a high concentration with the help of some transporter proteins. Therefore, it does not require energy that it procures from ATP. An example of active transport is the reabsorption of salts at the level of the renal tubules, or in the nerve cells where sodium goes outside the cell, where there are already medium sodium ions. The rate of active transport is directly proportional to the number of transport proteins. Cytosis. This can be exocytosis or endocytosis. Exocytosis occurs when a membrane-bound cytoplasmic vesicle fuses with the cell membrane and releases its contents outside the cell. Examples of exocytosis are the release of neurotransmitters by nerve cells and the secretion of hormones or mucus by endocrine glands. Endocytosis occurs when the membrane encloses substances and draws them into the cell through vesicles bounded by the membrane. It can enclose liquid or solid substances. When it encloses liquid substances, it is called pinocytosis. When they engulf solid substances, it is called phagocytosis. An example of endocytosis is when white blood cells remove microbes from the bloodstream. With that said, we come to the end of this video. If you have any questions, please let me know in the comments or on Instagram. I would like to know what you thought about this video. If you liked it, don't forget to press the like, share and subscribe button. Have a nice week.